Hi, let's do balancing act day 5. Our warm up question today was taking a look at the nitrogen cycle and trying to identify these different processes or steps that are labeled by A, B, C, and D. So I asked students to go ahead and get their notes out. You want those notes on your toy cars, and also, you want to have notes out for your balancing act 2.4, and we're going to do some things with those notes as well. All right, so looking at A, B, C, and D, if we want to name the process that's happening at each one of these spots, luckily it's right here on your form, on your diagram here. I'm looking at this blue arrow that turns nitrates back into nitrogen N2, nitrogen gas that's in the atmosphere at spot A. That's going to be denitrification. Then when I look at B, if I follow this blue arrow is going from nitrates to the plant's roots. So what's going to happen there? The roots of the plant are assimilating. That whole plant is assimilating the nitrates. So that's called assimilation. And when I look at C here, I am going from ammonium to nitrites. That's called nitrification. I'm turning something into a nitrogen compound that is not nitrogen gas, right? A nitrogen compound. And that's called nitrification. Whoops. And I gave you the answer already for D. When we're looking at D, we are turning nitrogen gas here all the way from the top. See, starting at the top. Follow that blue arrow all the way down. If we turn nitrogen gas into ammonium, that is ammonification. All right, and just a reminder that you have a test coming up. Your test is on nitrogen cycle and the experimental design diagram. And we gave one extra class day, so we are now giving that test on for even periods Tuesday, February 6th, and for odd periods Wednesday, February 7th. All right, quick reminder in class about fire drills. When we have a fire drill, immediately get silent, please. If we are evacuating, which we should when there's a fire drill, you should slide in your chairs, stay with your class, face away from the building once you're outside. Be silent out there while you wait for further instructions. And then when walking in and out of the building, you need to also be silent because you're listening for important instructions. All right. What are we learning today? We're going to continue to review the nitrogen cycle, and we're going to practice what we have learned about the experimental design diagram. Why are we doing that? We want to understand how nitrogen moves throughout an ecosystem. Nitrogen goes in and out of biotic and abiotic elements of, I shouldn't use the word element, it might be confusing, through biotic and abiotic sections of the ecosystem. And we also want to remember how to set up a proper experiment. All right. So thinking back to Mr. Lee's cat, we talked about this at a different class. His cat, Hallie, is not sleeping very much. So Mr. Lee wants to see if changing his cat's bed would affect the amount of sleep she gets. So if he's going to do some experiment, what is the one thing Mr. Lee will change here? He wants to see if changing his cat's bed would affect the amount of sleep. So changing the cat's bed, right? Then, what is the dependent variable? What is Mr. Lee going to measure or observe? He's going to measure or observe right here the amount of sleep the cat gets. Okay? All right, if we had to write a hypothesis, we might say if we change the type of cat bed, then the hours of sleep will increase. If the cat bed is changed, then the hours slept would increase. All right, here's another quick scenario just to get you thinking back about your independent variables and your dependent variables and hypotheses. My plant is not growing as fast as I thought it would. I would like to see if adding more fertilizer would affect the growth of my houseplant. So if I'm thinking about that, what is my independent variable? What's the one thing I will change? I want to see if adding more fertilizer. That's what I'm going to do. So it's the amount of fertilizer. Then, if I need to figure out what is the dependent variable, what am I going to measure or observe? How will I know if there was a change? The thing I'm going to measure is the growth of my houseplant. Is there going to be any effect on the growth of my houseplant? And I'll measure that, and that is my dependent variable. Sometimes we call the dependent variable the variable that responds to our independent variable, whatever it is that's responding to our change. So just keep that in mind. Generally, dependent variable is going to be data that you're collecting or you're observing or you're measuring. Okay, so next, if I want a hypothesis for this, I might think that if I add more fertilizer, then the plant will grow more. If I increase the amount of fertilizer, then the growth of my plant will increase. Okay, so in the classroom, I asked students to use a laminated experimental design diagram sheet and try to fill it out based on this scenario. So where Ben lived, he could hear crickets chirping in the late summer. Ben hypothesized that the hotter the day, the more often the crickets chirped. He designed an experiment to test his hypothesis. He built a cage of wire and wood and put some grass and leaves in the bottom. A thermometer was hung in the middle of the cage. He controlled the temperature of the cage with a pet heater. So Ben went outside and collected 10 crickets and put them in his cage. He did the tests over several days at 25 degrees Celsius, 30 degrees Celsius, and 35 degrees Celsius. 3 trials were done at each temperature, and for each test, Ben counted the number of chirps for 3 minutes. So first, I just want to remind you that room temperature is roughly 22 degrees Celsius. Our human body temperature is about 37 degrees Celsius. So if you think of that in terms of Fahrenheit, we are 98 or 99 degrees Fahrenheit. This is getting pretty toasty, right? So let's think about all of these thoughts and try to fill out the experimental design diagram. If I want to make the first thing I'm going to ask you to figure out is what is the independent variable? What is Ben going to change? Ben hypothesized that the hotter the day, so that was his thought, if I change the temperature, then the more often the crickets are going to chirp. So his independent variable is temperature. Sorry, there it is, temperature. And he did test 3 different levels of temperature, right? He tested at 25 degrees Celsius, 30 degrees Celsius, and 35 degrees Celsius. And that scenario said that he repeated each temperature 3 times. All right, so our independent variable is the thing that we are changing. Our levels are how are we changing that thing. And then our dependent variable is going to be what are we measuring. He's measuring the chirps of those crickets, right? So we want to think about constants. And the things that are going to stay the same, we're going to use 3 minutes as our timer for when we count chirps for every single situation. We're always going to use the same thermometer. We're using the same type of cage with the same stuff inside of it. And so we're using leaves and grass. We are using the same types of crickets, and that's important because different species of crickets chirp at a different rate anyway. I didn't mean to have same type of crickets on there twice. Sorry about that. And if I wanted a title, I might say the effect of hotter days on cricket chirping, or the effect of temperature on the number of chirps by a cricket. And we are guessing that if it is hotter, then the cricket will chirp more. If it is hotter, if the temperature is higher, then the amount the crickets chirp also is higher or increases. All right. So think back to your toy car experiment. And because some students weren't quite able to finish all of their trials in our last class, we set up our experiments for 7 whole minutes in class. If you need to do more of these runs with your toy car, you can come during Falcon Focus, or you might be able to get the additional data from one of your lab partners. Okay, that was 7 minutes that I had people work on that. All right, so when we came back in, we were getting ready to talk about what we're going to do with our data. So we have to just think about experimental design in general. The first thing we do when we're setting up an experiment is we have a question. What kind of a question do we have? What are we trying to solve? In this case, we are trying to figure out what impact does ramp height have on how far a toy car travels. That was our question. And so we wanted to set up a fair or valid experiment. What is a fair or valid experiment? It means it's a fair test. I'm not setting it up with, I guess you might say, faulty equipment or something. I don't have dice that are weighted on the bottom. I'm using appropriate measuring that's true and valid. So also we're going to test what we said we would test. So in our case, we tested how far the car went after we changed the height of the ramp. And for all appropriate and validly designed experiments, we can repeat the experiment and get close to the same results. So that's why we do extra trials, but somebody else ought to be able to come up and do the same exact type of thing you did and get similar results. Okay, so you already had set up your experimental design diagram in class before you started working with your toy car. Once we work with the toy car, we need to collect data. We need to collect data and then do something with it. So a lot of times you're going to have a data table, which could be a spreadsheet. It could be an online spreadsheet, or it could be a paper spreadsheet. So we had a data table that had the number of books we used for our ramp, and for each trial, we had a different cell in our table. Okay, so we collected the data, and then what we need to do is average the data. So if I had 0 books as my ramp, or my ramp height rather, I would add all 3 numbers that I got on my 3 different trials and divide by 3. So I'm getting an average, right? So if I had done 4 trials, I would add all 4 numbers together and divide by 4, right? All right, so back in class, I asked everybody to go ahead and average those columns. And then you're going to analyze or draw conclusions. So we're going to analyze the data and draw some conclusions. If you had already done your graph before you came to class, I know some people might have chosen to do a line graph. We chose to do a bar graph in class just to show this data. And so when we are setting up our graph, always your independent variable is going to be the x-axis, this horizontal axis. Okay, that's what you are changing. So we were changing the number of books that we were setting our clipboard ramp on. The dependent variable on the y-axis is always going to be the thing that you're measuring. It's how your test subject responded. What did you measure? So in our case, again, x-axis was ramp height, or the number of books, and y-axis was the distance the car traveled. Be sure that you put your units. So in this case, we use centimeters, and typically scientists are going to use metric. Okay, we're going to use a bar graph, so I'll just remind you about what bar graphs look like, and then you'll analyze your data. All right, so an example would be, here's an example. An example would be if you asked a bunch of students what was their favorite musical instrument. Okay, so this is a graph that shows what was the favorite instrument for a bunch of students. So what we need to put on our graph to make sure that other people understand it, we need a title. So you'll need a title on your graph. We need to have our x-axis, the thing we changed. This is the independent variable. And our y-axis, and this one is the number of children. Those have to be labeled. They have to be labeled so that people know what they're looking at. And we need to have some sort of a scale. So you are going to decide what your scale is depending on the numbers that you got when you averaged all of your trials together. Okay, so you're going to look and see basically how many boxes do you have on your graph that's on your worksheet, and you might go by 5s, or you might go by 10s, or you might go by 20s. You're going to see what scale you need to represent your data. All right, and then you need some sort of label that describes your categories or your bars. So for us, we're going to have 0 books, 1 book, 2 books, maybe 3 books, depending on what you did with your group. Okay, I gave people a couple minutes to get that done, and then I reminded everybody that when you turn in your work, you need to have your first name and your last name in order for me to give you credit. If I don't have your first name and last name, I might not know which student you are. If you write your name in a way that I can't read it, I can't give you credit, right? Because like looking at this was an actual example from something that somebody turned in. I don't know who to give points to for this lab, even though it was done fine. So please, if you want credit for your work, tell me who you are. And first and last names, full names. Don't just give me your initial unless that's all you have. Okay, so after everybody analyzed their data, and sorry, this is an additional slide for that same information. On the bottom left of your worksheet, there's a question for you about, was your hypothesis proven? You need to think back to what your hypothesis was on your experiment, because it was your experiment. Most people in our classes probably said, if we increase the ramp height, then we will increase the distance the car goes. So if that was yours, then you can think through with me. Otherwise, you need to think about your own hypothesis and whether your data showed that you got it. There's a spot on your page to write down what you learned. Okay, what did you confirm your hypothesis is what it says. So if you did confirm it, you would say, if ramp height is increased, then the car distance also increases. You just kind of turn your question back into a statement. All right, so people turned in that lab sheet, and then we did a little bit more in-class practice. So I'll go through a couple more slides, and then you'll have some independent work to do. All right, so think back to the carbon cycle. Of course, right now we're doing the nitrogen cycle, but think back to the carbon cycle. When we think about the carbon cycle, we can think about how humans impacted this cycle, right? We know that humans burned a lot of fossil fuels. We still do in cars and factories and things like that. So that's one thing that humans have done to impact that carbon cycle. So we're going to actually apply some of our thinking process to what happens with a nitrogen atom. We already did. Let's hop a ride with a nitrogen atom, but we did a nitrogen atom through natural events, lightning and decomposition and water runoff and things like that. So now we're actually going to think about what happens to nitrogen as a direct result of human activity. So you're going to go back into Schoology and you're going to follow a virtual simulation. So you're just doing this on your computer, and you're going to, I'll back right up. You're going to put this all in your travel log in your packet. This is why you need your Balancing Act 2.4 packet. So just a moment ago, I said, how did humans impact the carbon cycle? We burned all those fossil fuels, and we have released excess carbon dioxide into the atmosphere. So the sphere that was the most affected by our changes in carbon, the sphere would be the atmosphere. All right, I have a video here that I will come back and play in just a moment. I'd like to work through the nitrogen cycle first, and then we'll come back to that video. All right, so you're turning to this page in your packet. And we're not going to put anything in this first cell because we are starting in fertilizer, and we want to know, if you start in fertilizer, which form of nitrogen are you? So when you go into Schoology, you're going to see this virtual nitrogen cycle journey. There's a link for it, and you'll open it and you'll click me to start, just like it says right there. You'll click, that's a link. Okay, nothing goes in this box. Everybody is starting as fertilizer. So when you click, you are starting as fertilizer. So it says right here, you are the ammonia, nitrites, and nitrates in the fertilizer. So that's the form you've taken. And you can either write it out as NH3 and NO2 and NO3, or you can write the words ammonia, nitrites, and nitrates. Okay, so you can fill in that box. Then it says, which location are you moved to? Well, it turns out we started as fertilizer and we got moved to soil. When we rolled, our first example was a 3. So I just had everybody go ahead and pretend they rolled a 3. And so we ended up in soil so that we could start filling out our form together. Okay, you were the form ammonia, nitrites, and nitrates. And what happened to you? Well, a farmer applied you were in fertilizer, right? A farmer applied fertilizer to their field, and then you as a nitrogen atom seeped into the ground. Okay, you seeped into the ground and you're in the soil. The thing is you're still the same form of nitrogen. See how it says unchanged? You're still the same form. You're either ammonia or nitrites or nitrates. All right, so what do? You roll again. Okay, we're going to roll again, and then you're going to fill out your whole travel log. I gave everybody 22 and a half minutes to work on this, but probably you don't need 22 and a half minutes for this journey. I have some other things for people to work on as well, so I'll click forward and we'll talk about those. All right. While you were working on your nitrogen cycle, you wanted to finish that. As soon as you did all of those steps, there was an exit ticket also in Schoology, so I asked everybody to do that exit ticket. If you don't get perfect score the first time, try it a second time. And when you've done both of those things, there's a formative on experimental variables that you can do as well. That formative, if students didn't finish it in class, that became homework. All right, so we're not going to wait 22 minutes on this video, but we are going to back up and watch the other video right here. So I'm going to turn off my voice and turn on the computer voice. I wasn't able to get my captions to show up for this. So make sure that you have the Google captions on if you're watching this at home or even in school, but you can turn on the Google captions. I couldn't get the captions to turn on for it. All right, here goes.
Hey, look at those weird bumps on the roots of that soybean plant. Bet you can't guess what's inside. Bacteria. Bacteria? The stuff that makes you sick? Yuck. Hey, not all bacteria is bad. In fact, we couldn't live without the bacteria in these little nodules. Neither can animals or plants. These little things help keep us alive? How? Turns out they're VIPs in the nitrogen cycle, the endless process where nitrogen moves back and forth between living things like plants and animals and non-living things. Like air, water, and soil? You got it. Okay, so all living things need nitrogen to survive, and there's lots of it in the air. Can't we just breathe in the nitrogen we need? Nope. Our bodies can't use it as a gas, and neither can plants. That's where our bacteria buddies come in. Do they do something to the nitrogen? Yeah. First, nitrogen gas travels from the air to the soil and right into those nodule thingies. Where the bacteria are waiting? Yep. The bacteria turn the nitrogen gas into ammonia, like the household cleaner. Cool. Then plants use the ammonia? You'd think, huh? But most plants can't. It's pretty toxic. That's where other bacteria come in. The ammonia leaves the nodules, and then bacteria in the soil turn it into nitrites. But plants still can't use the nitrites, so different bacteria turn the nitrites into nitrates. The whole process is called nitrification. Nitrification. Sounds royal. We dub these Sir Nitrate. Nitrates do play a royal role. Plants use them to create the proteins they need to live and grow. And animals do the same thing. So if plants get nitrates from the soil, how do animals and people get them? By eating plants. And animals that don't eat plants eat other animals that eat plants. How carnivorous. What about the rest of the cycle? How do the nitrates turn back into nitrogen gas? Plants and animals die, And animals excrete waste. A lovely thought. Everything decays and breaks down, thanks to special bacteria called decomposers, which release nitrogen back into the cycle. They reverse the whole process. And the gas ends up back in the air? Most of it does, and the whole cycle repeats. I think I've got it. Nitrogen is a gas in the air. Then bacteria in plant nodules and in the soil change it into nitrates. Next, plants and animals use the nitrates to make proteins they need to grow. And last of all, bacteria change the nitrates from waste and dead matter back into nitrogen gas. And the cycle starts all over again. Perfect. But what's not so perfect is that people are doing things that upset the cycle, like driving too many cars. That builds up nitrogen in the air and can cause smog or acid rain. Kinda scary since we're breathing that air. Can we help? Well, we can start by riding our bikes more. Now that's what I call getting to the root of the problem. I'm with you all the way.
All right, so I'll actually include that link in Schoology, just in case you want to take their little quiz that was online. So you have presumably gone through and done the human impact nitrogen atom travel log. You have finished your, let me get myself there. You have finished your exit ticket and you have finished your formative. So thanks so much for checking in. Remember, we have a test coming up and we will work one more day. We will have one more day of balancing act to practice, and I'll get a study guide to everybody during that class. Thank you.